So we can start now.
Will there be anything else before I start? Any introduction or something, or I can start?
No, sir, you can start.
Okay. Good evening. Can you all hear me? Can you hear me?
Yes, sir, we can.
Okay. This is the beginning of the mechanobiology lecture, and there will be 3 such lectures. So it's titled Mechanobiology, Principles and Application, and this is the first lecture. So the plan will be the following. I'll finish the lecture in time and keeping time for you to ask questions if you have any. And then you can type your question in the chat box, which I can then reply at the end of the presentation. So in the middle of the presentation, it's better to chalk out all your questions if you have some, and then ask them at the end of the lecture. Good evening to everybody, and I am Kaushik Sengupta from Biophysics and Structural Genomics Division, Saha Institute of Nuclear Physics. I thank HBNI and SINP for putting together such a wonderful coursework, or such a wonderful series of lectures, I would say, and that enables you to learn many new things. Mechanobiology is one of the very recent areas which is receiving increasing interest from the researchers throughout the globe because of the problems and the questions it addresses. So the lecture plan will be the following. In the first lecture, I would cover the components involved. And by components, I mean the components of the cell, which means the organelles of the cell, which are involved in the mechanotransduction. I will explain the term mechanotransduction in a few slides. The components would also mean the proteins, the lipids, whatever, which are involved in the process of signal transduction, which is stimulated by mechanical cues to the interior of the cell, from the exterior of the cell, that is extracellular matrix, to the interior of the cell. And then the next 2 lectures would be on measurement and applications. Measurements would mean a type of experiments that are typically used to address the problems which can answer the questions related to the response of a cell to mechanical force. So we will see how the experiments are designed, what are the mathematical principles, what are the physical principles behind the experiments, and what are the applications that we'll see in the next 2 lectures, which are on 17th of November, and the last one will be on 22nd of November. So what is mechanobiology? This might appear to be a very new term. So mechanobiology contains, as the name suggests, an element of mechanics, an element of biology. So this is one way of saying that it is the application of mechanics in biological systems. For what? Why do we need to apply mechanics in biological systems? So the answer is mechanobiology deals with the responses of cells/tissues to mechanical forces. In other words, it stands for events at the interface of physical forces and subsequent biochemical reactions. Here we can remember the famous line quoted by Charles C. Thomson, one of the polymaths who pioneered in biology, mathematical biology, and his magnum opus, Growth and Form, is still an authoritative book on the morphogenesis. And there he says, the fact that the germ cell develops into a very complex structure is no absolute proof that the cell itself is structurally a very complicated mechanism, nor yet does it prove, though this is somewhat less obvious, that the forces at work or latent within it are especially numerous and complex. Well, this sentence or these lines might appear to be a bit complicated to you, as it was written at the beginning of the 20th century. But essentially, the meaning remains the same, that the forces or the study of the forces at work at the interface of the cell and the extracellular environment has also a role to play, other than the complex mechanisms which are going on inside the cell, in the development of a whole animal from the germ cell. Okay, so we will study what are these forces, how these forces are dealt with in the biological system, how these forces are sensed by the proteins in the cells, and then how the cell responds to these forces. Mechanobiology comprises of two basic processes, mechanotransduction and mechanosensing. Mechanotransduction refers to the transmission of external mechanical forces inside a cell/tissue to elicit a biochemical response. Now, what can be an external mechanical force? Well, there can be many. There can be friction with your skin. It can lead to the tissue damage. As a result, you know that if there is an abrasion of the tissue, the wound will be healed in due course of time, and that is also due to some biochemical response from the cell. If the cells of your skin, which are of course exposed to the environment all the time, gets mechanical assault from the environment in different forms, it responds to it in different way. It can show hardening. It can break. It can have many other changes, and as a result, it will elicit the biochemical response. And then mechanosensing it pertains to a particular organelle or protein inside the cell. That's what I was saying, that these forces will be sensed. So these proteins or these organelles are the sensors which would respond to the mechanical cues. Okay. The development of mechanobiology, it dates back to 4th century before Christian era, when it was mainly the interest in biomechanics. Philosophers at that time, starting from Aristotle and physician Galen, they were all interested to find out the inner layer of understanding of the biomechanics by which we move our hands, move our feet, move our head on the neck. And that was the beginning of the biomechanics. That was the interest that was triggered by Aristotle and Galen. It was followed up on a more systematic manner when Leonardo da Vinci, the polymath of Renaissance, of uh, the all-time famous painter and mathematician, a polymath to be precise, he studied or started studying the muscle forces, the joint functions, flights of birds. And you might know that his study and his sketches on the flights of birds enabled him to design a flying machine, which is a very popular matter of fiction. And also this can be found in Da Vinci's notebooks. And that was the beginning of scientific application of study of forces, the joint functions of the human body, not only human body, of animals, to formulate a mathematical regime where these forces can be quantified and the response to these forces can be quantified. It was followed by Galileo Galilei, who went a step forward and devoted much of his time in studying the bone development, bone mechanics, bone mineralization, so on and so forth. Ultimately, we had to wait till the 17th century. Physician, physiologist Giovanni Borelli, he defined the term the tissue elasticity, muscle function, etc. And this is one of his this page is a very famous illustration of his book De Motu Animalium of Giovanni Alfonso Borelli. And here you can see that the bones have been placed in a manner that they can be studied as a, they have put a fulcrum and the weight and so on. I mean, whatever the people by that time have gathered about the knowledge of the weights, the lifting of weights, the force involved in that, these things were applied in studying the movement of the bones, muscle tissues. And this was the classical beginning of the biomechanics. For this lecture or series of lectures, we would focus on not biomechanics as a whole for the entire animal, that is in the case of the human beings, but we would see, or we would break it down at the level of the cells, because you all know that cells of similar type, they make up the tissues. So there can be epithelial tissues, there can be muscle tissues, connective tissues, so on and so forth. 4 types of tissues, including the nervous tissue. These tissues make up the organ, and the organ make up the body. So if we break it down to the simplest unit and try to understand how the cellular mechanics is controlled, that is the movement of the cell from one position to another. The classic example you can imagine is that of the metastasis of cancer. That is, one has the lung carcinoma, but if it's a very aggressive type, it can spread to head and neck, or if it's something related to your brain, it can spread to the different parts of your body. How this is possible? This is possible due to the fact that the cancer cells are aggressively dynamic in their motion. So this course would enable you to formulate, or more precisely, to visualize this movement, to quantify these movements at the level of the cells, at the level of the proteins, that is what changes the proteins are undergoing, and at the level of the deformation of the cell also, which largely depends on the elasticity or plasticity of the cell. We would define these terms progressively as we move on. But first, let us see the cell. This I've drawn the cell, the static cell, the first half of the picture here. The cartoon, the static cell, this is a cell. If you see it from the side view, and the major part of the cell is taken up by the hard nucleus. The nucleus contains these colored domains of the chromosome. Now we know that chromosomes are not just randomly oriented or distributed, but they are associated as territories. And the nucleus, like other organelles, which I have not drawn here for sake of simplicity, are held in a cytoskeletal network composed of actin, that is given by the brown line here, that is the thinnest of the cellular cables or filaments, intermediate filaments, IF is given by this orange line here, orange colored line here, and the microtubules, the green ones, which are the thickest ones. These cells, while they are static, they are anchored to the extracellular matrix via the integrins, and they are connected to each other by cell-cell contact. I will not go into the details of the names of these to complicate your life, but just understand that the cell, when it is static, it is some sort of attached, anchored, or they are actually placed in a fixed orientation on the extracellular matrix. And when the cell decides to move on response to force or chemoattractant, whatever, the cell, first it should detach itself from the neighboring cell, and it should also detach itself from the extracellular matrix, right? It's quite understandable. I don't need to explain, because you have to lift your foot from the ground to move. You cannot stand on the ground and continue moving. It's the same thing. So these are the feet of the cell. These are integrins, part of the focal adhesion complex. I will come to each of these slowly. And the cell, if it moves in this direction, given by the arrow front end, this is the rear end, it starts forming projections like this, which propels the cell in this direction. And these projections are called Lamellipodia. Comes from the Greek word, from the Latin word, the podium, which means foot. So basically, they're the foot of the cell, and they move in this direction as if they crawl on their foot in this direction. What happens here? There is active actin polymerization, bundling here in this phenomenon called Lamellipodia. Inside the Lamellipodia, there are fine projections, which I will show in the movie in the next slide. So if you see this thing, the cell, it's a NIH3T3 mouse cell. These membranes, this is called a ruffling of the membrane, okay? And you see these spikes coming out. These spikes are called Philopodia. And this big ruffled membrane, they are the Lamellipodia, okay? And so these actually determines the direction of motion of the cell. So if you see this thing, I can start playing it again. So see these projections, they are all called Lamellipodia. The finer needle-like projections inside these big projections, they are called Philopodia. Okay, this is the nucleus, and you cannot see the cytoskeleton here. This is a DIC image, differential interference contrast, and this is a live cell imaging, okay, by which you can see many of these nice images. If you go to this site, it's all given here. It's www.microscopyu.com. It's from Nikon, and this is a wonderful site where you can go and you can find all these images. All right, so this is the cartoon of a cell, and the cell is, I have not drawn all the other organelles, just not too complex matters, but I have just drawn the systems which are involved in mechanotransduction, or the active components of mechanobiology. Okay. Am I audible? Am I audible? Okay. So you have the nucleus here, and it is surrounded by surrounded by a network of microtubules. Just check whether it is audible or not. Okay, so you can see this thing. All right. These are the thickest cables are the microtubule. Actin represents these fine filaments here, and these green ones represent the intermediate filaments. And they actually they stretch the plasma membrane on the cytoskeleton. So these constitute the cytoskeleton, such that the cytoskeleton acts as a scaffold on which the membrane is stretched like a tent. This is the nucleus. Intermediate filaments are of many types. I will come to that. They are all cytoplasmic in nature, excepting there is one nuclear intermediate filament given in red underlying the inner nuclear membrane. It is called a lamin. These blue guys are the nuclear pore complexes or nuclear pores through which the nucleocytoplasmic transport goes on. And this double membrane bound nucleus protects also the chromosome. Okay, so this is the biggest organelle, and this plays important roles in the mechanotransduction, in maintaining the cellular rigidity, in maintaining its own rigidity, such that the chromosome conformations can be kept in the proper form. So it represents the tent, okay, the tent of a circus, which we have all seen in our childhood sometime or the other. The model of the cytoskeleton as proposed now is called tensegrity. The man behind the tensegrity, his name is Buckminster Fuller. You might have heard his name. The compound fullerene is named after him. And this is a ball made up of struts and springs. And these are all pre-stressed, okay? Such that if you deform any part of the ball and then remove the deformation force, it returns to its original shape. So it's a self-sustaining integrated structure. The term was coined by Buckminster Fuller, and he was a famous architect. And this term means the tense and gritty come from the two words, the tensional integrity. So, which means again, this structure is in a state of tension, which maintains the integrity of the structure. Similar situation occurs inside the cell, as explained by Don Ingber of Harvard through his pioneering work. We will see in lecture 3 the wonderful applications of the tensegrity model in the tissue engineering. But before that, we have to understand all this phenomenon before that. And I'll show you another structure of a tensegrity model by Kenneth Nelson, who's also a famous architect and student of Buckminster Fuller. That is, these are all these, these metallic struts, and these are the cables. So if you just fiddle around with this strut, it will go back to its original conformation because these springs are all stressed. Okay, so again, the term, the tensional integrity. Please remember this term because we will refer to this term back and forth. So based on that, it was Don Ingber who built several of these models. And as I mentioned, he's a pioneer of this school of or this school of thought that tensegrity maintains the tissue integrity, the cell integrity. So this is just a simple, like a matchstick connected with some elastic bands. And if you distort if you just pull at one end, let it go, it will come back to this form. Similar with the springs. So if you connect, the system of springs, they interconnected. If you just distort one spring, let it go, and it will come back to its original form. So you can refer to these references, which are given at the bottom of the slide for a detailed understanding of this thing, which is beyond the scope of this limited time of the lecture. So here is a cartoon. I took it from Adhesion of Biomed Communications. So this is actually what I was mentioning before, that you actually pull this thing at the top, okay, and see what this guy does. So you pull it at the top, and then it comes back to its original form. You pull it from the side, and then it undergoes some sort of deformation, and then again it comes back to its initial state. Okay? So this is the visualization of the tensegrity model. Now, it's a very important slide. It's a busy slide, but don't worry about these things at the bottom. These are the references. This lecture will be visible, will be available at the YouTube. Anytime you can go back to these papers, and that's why these references are very important. So here, what is meant, it's a cartoon, and suppose the shear stress is given on the cell. This is the cell in this direction, right to left, and the cells are in contact with the extracellular matrix given by ECM, and the elasticity of the matrix, the mechanical, the strain, it increases from the right to the left. Okay, so as a result, what happens, there will this force, this shear force, that is the deformation of the cell. If you can imagine, if you just put your hand on the cell and you just squish it like this is the same thing what is given here. And then what happens is the cell will be deformed. Okay, and as a result, all these molecules, the rho GDP, rho GTP, rock, and all these things which are involved in the remodeling of this cytoskeleton will be differentially regulated. As a result, the the force sensing sensors, that is these actins, these microtubules, the nesprins, they will adjust to this force in a manner that this information of this force is passed to the nucleus, okay, to the lamina. And this represents a gene expression with this arrow. Why gene expression? Because if the nucleus is also distorted, if you can imagine, you distort a cell, you shear a cell in this direction, so the nucleus will also be flattened. Okay. As a result, there will be some shearing stress felt on the nucleus also. As a result, the chromosome territories would also be affected. And as a result, the genes on the chromosome would also be affected in their expression. And there will be transcription up regulation or down regulation of genes, which would act in a feedback loop. This is the most important part, please understand. This will act in a feedback loop to resist that deformation, to bring the cell to the original conformation. All right? And this constant stress on the cell, starting from the germline, is the basis of the differentiation into so many lineages. And that's why when we have the zygote, the embryo, from that little thing which is implanted in the placenta, it's a mass of a ball of cells, we have the entire organism with well-differentiated organs, which each perform their different functions and contain different tissue or cell types originate. Okay? So as I mentioned, what are the tissues of the body? So the tissue is a group of cells that we know that perform a similar shape, have a similar shape and function. Different types of tissues can be found in different organs. There are four basic types: epithelial, connective, muscular, and nervous. This epithelial tissue covers the body surface and forms the lining for most of the internal cavities. The major function of epithelial tissue includes protection, secretion, absorption, filtration. And as you know, skin is the largest organ of the body. It consists of epithelial tissues of different types, and cells of the epithelial tissue have different shapes. Cells can be thin, flat to cubic. Then there are connective tissue, which is the most abundant and most widely distributed of the tissues, performs a variety of functions, including support, protection. The following tissues are found in the human body: ordinary loose connective tissue, fat tissue, dense fibrous tissue, cartilage, bone, blood, lymph, which are all considered connective tissue. Likewise, there are the muscle tissues, skeletal, smooth, and the cardiac muscles. The skeletal muscle is a voluntary muscle, smooth muscle is found in the walls of the internal organs of the blood vessels. They are of involuntary type. So there are the different classifications of the muscle tissues. So there are the muscle cells making up the muscle tissues. The connective tissues are made up of the fibroblasts of the mesenchymal cells. The epithelial tissues are made up of the epithelial cells. The nerve tissues are made up of neurons and these different types of cells. Now, we see that the different types of epithelium here have evolved due to the requirement of the human body. For example, if you have the squamous epithelium, the squamous epithelium is present in the very smooth inner lining of the buccal cavity, in the cervix, very flat tissues. The cuboidal epithelium, which are mainly present at the inner lining of the organs. Cuboidal, columnar, present in the intestine. So each of these tissues have evolved based on the environment where they are meant to be. Now that environment means the different shearing forces that these cells have to undergo. Now if you see the blood flowing through the endothelial vessels, this is the endothelial cells, they are exposed to constant shearing force of the flow. So wherever there is a flow, there is a shearing force. All right. We will come to that in the lecture 2, when there will be some mathematical descriptions of the shearing forces and the response to that. But right now, what I am emphasizing to say that these cells have evolved based on the force spectrum to which they are exposed to in that atmosphere, in that environment, in that microenvironment to be precise. Okay. And we can understand it better if we see, or if we compare, when you go back home today after the lecture, or if you are at home, I am pretty sure you've always touched the heel, and you know that the heel, the mass of cells here, they are rough. They are not as smooth as the cells of your cheek. Okay, why? Because your foot, your heel, they are in constant friction with your shoe. When you're walking on the floor, they are in constant friction. So if you're not lying down, so when a baby is born, you will see that the bottom of the foot is as soft as the rest of the body. It's very soft. But when it starts to walk, when it becomes a toddler, after crawling, it starts to walk, and gradually, when the baby attains adulthood, you will see that this mass of skin will be increasingly hard due to the forces. So this is a response to this force that the cells have told themselves that we got to be hard, we got to be resilient to overcome the shearing of the cells due to this force. Imagine if you would have or if you graft your heel with the skin of your cheek, okay, your heel will, will actually burst into blood, and it will break open the moment you walk a few steps, because these cells of the cheek, they are not meant to take so much of load as these cells have evolved. Now, what specialty have been inculcated into these cells as opposed to these cells? These are more keratin here, okay, as opposed to here. As your nails, right? They are full of keratin. That's why they are hard. Likewise, you see the elbow, you see the knee, you see the mass of cells here. They have a different color. They have a different sort of smoothness, which is much less smooth than your cheek or the other smooth parts of your body. That's the upper arm or your armpit, which are never exposed to any sort of shearing force like that. This is the image of the palm of a worker who works in the field, because he has to use his wheelbarrow, his other instruments for plowing, and that's why he is constantly exposed to friction, is constantly exposed to some forces, right? And as a result, if you touch the skin, you can say it from this picture that it's got to be hard. It is hard. So again, we come to this diagram just to refresh your memory, because this was a cartoon, and now we would really see what's really happening down there. Now, in order to understand the architecture of the cell and how it is anchored to the extracellular matrix, we need to understand, or we need to focus a bit on this little house, which is anchored, which has his foundation in the basement. So if this house doesn't have a foundation, what happens? The hurricane or tornado or a few gusts of wind, and this house just topples. So it has to be anchored to some sort of a matrix, some sort of a foundation, which holds it against some forces which are acting in this direction, right? So that it doesn't topple. Similar things happen to the cells. So imagine, just replace in your memory the house with a cell, and these are the foundation layers, and these are these, if you go to the previous slide, these things here, okay. So these are the integrin molecules, okay. This is a plasma membrane. Let's assume it's an epithelial cell, okay, your skin cell, and underneath there is this basement membrane. Now, this basement membrane, please understand, contains no cells. All right. I emphasize they contain no cells, but they contain a diverse array of proteins, of polysaccharides, of structural proteins, okay, which form a mesh on which the integrins are anchored. What is the force of anchoring? In the hydrophobic interaction, ionic interaction, Casimir forces, short range forces, van der Waals interaction, okay? Because this is all protein interaction. Deep down, there is another guy sitting, which is the fibroblast. Okay? This is the connective tissue. That's why we introduced the concept of the different tissues there. And this fibroblast, as the name suggests, so you don't have to memorize anything, just the name of the cell speaks for it, okay? Fibroblasts synthesize the fibers. What do they synthesize? Synthesize the collagen, one of the most abundant proteins of the body, okay? So the collagen is synthesized, they are secreted, they come here, these laminins, these elastins, all synthesized, they come in here, make up the basement membrane on which the cell is anchored or tethered. The extracellular matrix, now we will see the constituents of the extracellular matrix. It's composed of matrix structural proteins, which are mainly 20 different types, just roughly. And the most abundant are the collagens, matrix polysaccharides, the glycosaminoglycans, syndeckins, glypicans are the examples, and matrix adhesion proteins. Remember, excuse me, there are the two types of proteins I mentioned, structural proteins, adhesion proteins. Polysaccharides are attached to the proteins, so they are glycoproteins actually, okay? And these matrix proteins are directly underneath the cell, where underneath those adhesion proteins are the structural proteins, okay? It's like the piling. When you build up a structure, you need to do a piling with these iron structures at the foundation, and then on top of that you erect it. So this is a piling. By piling, you mean that you make a strong array of network fibers on which these matrix proteins, the glycoproteins, are attached, on which the cell sits as like a cushion, okay, and attached to those matrix adhesion proteins by integrins. So this is exactly the picture what I was trying to convey. So this is the plasma membrane, and these are the integral membrane proteins, that is the integrins, as the name suggests, integrins. There are many types. We'll not go into the details, but just the schematics, because the fundamental of this course is not to expect to memorize all these proteins, hundreds of different types of proteins, but to understand their role. And so the integrins, they are connected, you see, they are connected to this, the fibronectin, the laminin, the collagen forms this broad network. So this is the extracellular matrix, plasma membrane, this is the cytoskeleton. Here you cannot differentiate, or I have not, I have chosen a picture where it is not essential to differentiate between the actin, microtubules, or the intermediate filaments, but just I'm showing the skeletal network. Okay, so this is the same image scanning electron micrograph of chitosan. Here, chitosan is a polysaccharide. It's present in shell of the shrimps and the lobsters. So if in the chitosan you mix elastin, okay, and electrospin it, it's a device by which you make these fibers, you see that with increasing concentration, that is D is more than C, C is more than B, and in A there was no elastin. So these forms, along with these, the polysaccharide, a dense network. So this is just an in vitro proof of what is there in the cytoskeleton. All right. So as I mentioned to you, the elastin, as the name suggests, this is elastic in nature because of the structure, the many helical coils, and you can actually extend the structure. If you touch the skin of the baby, it's very gentle, at the same time it's elastic. Okay, so and if you touch the skin of an old person like this, you will see that the skin is wrinkled, and this is presumably due to less of elasticity. Many other proteins are also there which are downregulated, but elastin is one of the principal proteins which are downregulated with increasing aging. Okay, and these are the crystal structures of the laminin. As you can see, that these are the structures which consist of the beta sheets, or there will be alpha helical structures in some other proteins, which means that they can be extended. If you pull from one end, they can be extended. Okay. In the subsequent lectures, we will see the methods employed by AFM and the optical tweezer, how you can pull this protein from end to end and find out the persistence length or the elasticity of each of these individual proteins. We will cover that in lecture 2, presumably lecture 3 also. So these are all the structures, and to give you a schematic idea of how the laminin is associated. So this is the plasma membrane schematic. The laminin are present in this form, okay? And on top of this, the collagen starts to build up. And speaking of the collagen, here comes the collagen, it's a triple helix. All of you know this thing, and these are the these electron micrographs. These are the TEMS, transmission electron micrographs from the skin sections, which shows you the collagen. Okay, it's a very rigid filament so that it supports the entire load of the cells which are built up on top of the basal lamin. So this is the principal component, the basal lamin, the extracellular matrix per se. Now, what are the components? So long we were outside the cell. I'm sorry. So long we were dwelling in the extracellular matrix. Now we make our way inside the cell and see what are the cytoskeletal components, what are the nucleoskeletal components. So actin, intermediate filaments, of which lamin is a nucleoskeleton protein, microtubules, and link. It's an acronym for linker of nucleoskeleton and cytoskeleton. Li from linker and NC, nucleoskeleton and cytoskeleton. All right. We will study each of these and the functions of these. And in the subsequent lectures, lecture 2 and 3 on 17th and 22, we will see how each of these proteins can play diverse roles in maintaining the elasticity of the cell, and how we can quantify, how we can measure, how we can visualize the elasticity of the cell under different contexts. Function of the cytoskeleton: provides anchorage to the cell organelles, very important, imparts shape and rigidity to the cell, aids in the transport mechanism inside the cell, helps in segregation of chromosomes during cell division. You know, microtubules do that job. A bit of info about the actin and the intermediate filaments and microtubules. The diameter is 5 to 8 nanometers. That's a cross-sectional diameter of an actin fiber. If you take, there are semi-flexible filaments involved in the establishment of polarity, morphogenesis. Remember the polarity when I mean the Filopodia? They are protruding. And what are the filaments which are present there? It's actin, right? So actin polymerizes in the direction of the movement, right? Which means it has a polarity. Cell motility and the persistence length is something, it's a mathematical concept, but for now you understand that this is the minimum length along the protein fiber, along the fiber, which can be bent. If you can be bent before something, which means you are resilient, okay? You are compliant. So this guy, actin, is compliant to what extent? 3 to 17 micrometers. So it can be bent. Let's see who can be bent most. Next comes the intermediate filaments, 10 to 12 nanometer in diameter. As you can see, the diameter is between the diameter of actin and microtubules. Then that's why it's called intermediate. That's why the name intermediate. Although lamins have now been shown, which has now broken the paradigm, it has set a new paradigm for lamins, that they are 3 to 4 nanometers, which means even smaller than actin. But let's not go into the complication now. They are semi-flexible filaments involved in maintaining stiffness and rigidity of the cell. Persistence length, oh my God, that is 200 nanometers to 1 micron, which means that they are even softer, right? So it's a minimum length, it's 200 nanometers, which can be bent. Imagine in the eye of your mind that if you have a filament, you try to bend it. If you have an iron rod, is it easy to bend it? Or if you have silicon elastomer, it is easy to bend it. Okay, we'll get the answer. So that is the persistence length is more in the case of the silicon elastomer compared to that of the iron rod. Then comes the microtubules, rigid hollow cylinders. So long we were in the discussion of the filaments which were not hollow, but this is like a pipe, okay, through which the water flows. You've seen the pipes. So it's a rigid hollow cylinder involved in karyokinesis, intracellular transport, cell rigidity, persistence length 1 millimeter. Okay, so this guy is the most rigid cytoskeletal component of the cell. followed by actin, followed by intermediate filaments. I repeat, microtubules, more than actin, more than intermediate filaments. All right? This will have important implications in understanding the later lectures. So this is from one of our recent reviews. So we have drawn the cell which is devoid of all the organelles. Just erase it, all the organelles from your mind right now. That there is no Golgi, no ER, no lysosome, nothing. So because those are not involved in mechanotransduction, right? So we are just showing here the components which are involved in mechanotransduction. What are the components? Integrin, focal adhesion. There's a bunch of proteins here which act as a linker between actin and integrins. Intermediate filaments, shown here with the thicker bundles, they converge with the integrins by presence of the hemidesmosomes, which are a different bunch of compounds, the complexes, and the desmosomes are also present in maintaining the cell-cell contact. Okay? Now, actin and intermediate filaments, and these are the microtubules, they do not directly reach inside the nucleus. So the force comes here, you distort the extracellular matrix, or there is a shearing force in this direction, or you pull the cell in this direction, whatever. So this cell is, if you actually push it to your left, the cell moves like this. So as a result, there is a reorganization of the integrins at the bottom of the cell, and as a result, this signaling starts from here, that is transmitted through actin or intermediate filaments. So the cell is here, you move it like this, and at the bottom, that is here, the focal adhesions, they sense it, they're the sensors. They're transmitted through the actin filaments here, the red ones, but where do they go? Well, actins also continue inside the nucleus. The nuclear actins are also there, but for sake of simplicity, I have shown that another protein, the nesprin, which are the members of the link family, which finally transmit or relay the force to here. Okay. So this is actually acting as an actuator or something that's actually, it multiplies these forces. This protein has multiple spectrin repeats. So it receives the force from here, it multiplies the force here. Okay, it's basically a multiplier, and then it transmits the force to the nucleus. Nucleus gets deformed, nucleus gets information or an idea that something is going on, and immediately it reorganizes its chromosome. Okay, so the nuclear force sink is the lamin which is present underneath, okay, to which the chromosomes are tethered. So if the lamin is deformed, that is intermediate filament, which will be also deformed as a result of force transduction inside the nucleus, so will be the chromosomes. And as a result, what we will see, what we can expect is any change in the force would probably lead to a change in the gene expression inside the nucleus. Okay, I'm sorry. So this is the crystal structure of the actin, the helical structures. So this is called the globular actin, and when the globular actin forms a filament, it's called F-actin. These are the crystal structures. You can read these wonderful reviews by Tom Pollard. They were the pioneers of the crystal structure of actin, and those who are interested in the structure can read about that. But there is more beyond this. We are not interested in structure. We are interested in the function in terms of the force transducer. Okay, so hence from now on we would refer to this as F-actin. Inside the cell, the F-actin are called by different names: the microfilaments, actins, stress fibers. This is one of the data from our lab. This is an ovarian cancer cell line, which is this is stand for the lamin A, which is mainly at the nuclear periphery, and this is the actin, which is stained by phalloidin, a drug. It's the rhodamin tagged phalloidin, which gives this the red fluorescence when excited at 546, and then these represents the focal adhesions, okay, which terminate at the tip of the actin. I will show you more images of this, don't worry. And this is the merged image of this thing. So this is exactly how the actins are stretched in a cell. Now comes the intermediate filaments. As the name suggests, as I mentioned also, their diameter of the filament was known to be 10 nanometer, in between that of actin and microtubule. Now, intermediate filaments has no structure. Okay, this is a buzzword. Why? Because intermediate filaments have a grossly unstructured N-terminal domain and a grossly unstructured C-terminal. Excepting for laminae, the type 5 intermediate filament, the lamins, which contain a little structurally aligned motif in the C-terminal, but yet the whole protein cannot be structured, cannot be determined by X-ray or NMR. Huge size of the proteins, grossly unstructured C and N terminal domains, which prevents the crystallization, and also the NMR, that is the solution state NMR. They are of 5 types, 4 types, 1 through 4, which includes the keratins, the most abundant proteins of the body. 70 different types of keratins are there, acidic and basic keratins, type 1 and 2. They make the keratins. The vimentin, desmin, peripherin, GFAP, glial, fibrillary acidic protein, they are type 3. Type 5, the type 4 are the neurofilaments, the nestin, cinnamon, internexin, mainly present in the nervous tissue, and the type 5 present in the nucleus of all tissues. Present in nucleus of all the tissues. Please remember, lamins are present in all animals which has nucleus. They are present in the C. elegans, present in the Drosophila, present in the higher in the higher mammals, in the higher organisms, but they are not present in plants, although they contain a well-defined nucleus, and they are not present in anucleated cells. Okay. All right. So these are the cytoplasmic 1 through 4 are cytoplasmic intermediate filaments, which mean they contribute to the cytoskeleton only. Type 5 contributes to the nucleoskeleton only. So these are some of the images from my laboratory showing you the structure of vimentin, the type 3 of the human foreskin fibroblast. And you can see these wonderful fibers, these thread-like structures, which are sparse towards the periphery, but which are very dense towards the nucleus. That's the juxtanuclear region is very dense, which has of course its utility or its function, because it helps in the proper anchorage of this big organelle which is called nucleus. Okay. And this is the nucleus. This is also a super resolution confocal image from my lab, which is showing this wonderful network of the lamins, which are of A, B, and C types. Well, A and C are from the same gene. This alternate splice variants and different D proteins stained by the green and the red, which form a network. You can see the network, a well-defined network. And so this network is present underneath the inner nuclear envelope, and this network encompasses the chromosome, or this acts as a shield to the chromosome territories. It protects the chromosome, it protects the nuclear contents, and also maintains the nuclear rigidity and shape. So you can imagine that any mutation in these protein can be potentially detrimental, and they are so, because we have seen through our research that these intermediate filaments have the lamins. They have numerous mutations. Lamin A has around 500 mutations which are reported. They produce about 16 different diseases of the human body, and these diseases are called laminopathies. And the cardinal feature of the laminopathy is misshapen nuclei, that is, the nucleus blebs, and the network is broken at places, and this leads to cross transcriptional malformation. So this slide presents the Vignetting Assembly. These are the transmission electron micrographs. in vitro assembly, which means that these proteins you can prepare in your lab, and with these proteins then you can polymerize, and you can follow the polymerization through, from 10 seconds all the way up to 1 hour, which you can, and then after you can see in this grid, if you put it in the electron microscope grid, very tiny grids, if you put a drop on the grid, negatively stain it with uranyl acetate, and then or sometimes you can also rotary shadow it. There are the various techniques of seeing these filaments, widely studied intermediate filaments. You will see that it gradually forms a spaghetti-like structure here. Okay, and this is the from the fluorescence microscope, the image from the fluorescence microscope, again the different time points, showing that the tiny little squiggles, as they're called, these are the term used in the Vignetting literature, that are very tiny filaments which correspond to this gradually build up by end-to-end attachment, forming these filaments like this. Okay. Similarly, the in vitro lamina assembly has also been extensively studied. The major tool has been the electron microscope. Okay, so here the basic difference is the lamine forms a dimer first. It's a coiled dimer, and then it forms a tetramer, either anti-register or register end to end. There are all these combinations. I'm not going to that. Interested one can read this literature. And then instead of this sidewise association, which was prevalent in the Vignetting, the major form which picks up is end-to-end association. That is, they elongate. Okay, and then these elongated fibers, they have different names in the field as the jargons. The protofilament, the protofibrils, and ultimately they form the 10 nanometer thickness intermediate filaments. So if we actually see this thing in the electron microscope, this structure is called the paracrystal, and any sort of mutation in the protein we can study that it leads to a defective association of this network, which is proposed from these electron micrographs. You see this myosin head-like structure. This represents the coiled coil. Okay, this head represents the tail which stick out, which actually are the two balls which are completely unstructured, and then the dimers or tetramers, they associate end to end to form these long filaments. And if you allow these long filaments to polymerize for a long time in the polymerization buffer, it will form structures like this. Well, if you delete the head or the tail domain, you can still have association which can tell you that this association has deviated from the normal association like this. And this has numerous mechanobiological implications, which we will see later. We come to microtubules, which are formed of the alpha and the beta monomers. So it's a heterodimer of alpha and beta, and actually it is a hollow. It's a hollow coil. It is actually a hollow cylinder. Okay, and there are various studies on the assembly of microtubules and various phenomena called catastrophe, when it breaks down after it starts to build up after a certain length. We will not go into there, but we will only consider, or we will restrict ourselves to understanding its role in mechanobiology. So these are again the crystal structures, as you can see, these helical helical structures, which are the signature of all these cytoplasmic filaments. And this is the staining of the lung cell with the microtubule network. It's courtesy is the Olympus Life Sciences. And then we come to the focal adhesion complex, which I showed at the very beginning. Remember the integrin, and there was this focal adhesion I mentioned, which forms a connection with the actin. And you see these tips, these red tips, they are stained with any antibody corresponding to these proteins, which are zixin, vinculin, tallin, axidin. These are the antigens or the proteins which are present at the focal adhesion. Okay, so here these are the proteins here. Okay, this is a schematic from Keith Borit's lab, and they published it in FABSJ. It's a wonderful review, and you will find many articles there that this is a schematic view of the extracellular matrix. That is these integrins that's sticking out of the plasma membrane in contact with these basal lamina. And these are the proteins which are the pexilin, taline, vinculin, zixin. If you happen to have antibodies against this, against any one of these, you can stain it with this and you can see these lovely structures. And these are the actin stress fibers. Okay. All right, so these things are differentially activated, as you can see, that there can be activation due to the different sorts of the forces. That is, these integrins are the structure of the integrins, they are differentially modified. And in response to that, all these focal adhesion complexes, they are organized. If you want to study a live cell exposed to a stress, or some sort of shearing stress or some sort of stress, you can label, you can use the GFP or RFP or any fluorophore tagged variant of these proteins, okay, and express it, transfect it in the cells, and then you will see that you don't need to use this antibody, but if you excite it with a particular wavelength of light to excite the GFP or YFP or CFP or RFP, whatever, then those proteins light up and you can see a wonderful image of this focal adhesion complex. Next, we come to the link. This is the last candidate of the cytoskeleton. We have nearly come to the end of the lecture today. So link contains these are giant proteins. These are this is called the nesprints. And these nesprints contain the spectrin repeat, as it's the name. So these are the multiple spectrin repeats. Okay, remember the spectrin present in the red blood cell, might have all read about it. So these are the spectrin repeats. These are present in the cytoplasm. This is the red one is the cache domain. Okay, so this cache domain is in contact with the sun protein of the inner lumen between the outer nuclear membrane and inner nuclear. So the force is transmitted from actin to nesprin or intermediate filaments to nesprin via this domain cache. It is an acronym of CLAR, unschist, Sark homology domain, Syn homology domain, I'm sorry. So this is a small domain which is conserved in all different types of nesprins which are there. Different types of nesprins are there. So this force terminates in this cache domain. This cache domain is in contact with the sun protein. This sun protein is in contact with the lamin. I have shown you many times now the cartoon of the cell. Try to remember that, and you will understand how the force is relayed from one component to the other component. Okay, how the relay occurs, I'm coming to it in just a minute. And this is the staining. So using their antibodies, Bio USA and VWR. So there are both so this I mean to emphasize that is the nesprints are present both in the cytoplasm as well as in the nucleus. So there are both nuclear and cytoplasmic variants. Okay, now I was saying so long, we have come to the end of this lecture, that these forces are transferred, they're handed down from one component to the other. How physically they are handed down? So there must be the close proximity of the proteins such that the force, how the force is transmitted. It is not a relay race in the Olympic that you hand over the baton to the next guy, but these are the proteins. They have a different sensor mechanism. So the sensor mechanism is the stretching of the protein, the bending of the protein, right? So the stretching and bending of the protein, you will see we can use wonderful techniques of AFM, optical tweezers, and many other methods which are the tools of mechanobiology to find out the change in the contour length of the protein, the change in the persistence length of the protein, and by that the protein interacts with the next protein in the line, which also changes its local conformation. It then passes over the information by changing the conformation with the other protein. Now, how these interactions are taking place? Primarily, there are three types of interactions: hydrogen bonding, which can make and break. Of course, you can understand it's not a covalent bond, but remember, as a result of these forces, there can be covalent modifications. I did not show here, just to avoid complexity, to keep life simple, that when you have the focal adhesions as a response to force, focal adhesion kinases, FAKs, are some kinases. As the name kinase suggests, kinases phosphorylate. So they phosphorylate some proteins at the focal adhesion complex, which means these are post-translational modifications, or they have put the phosphorus group covalently on the protein. These modifications are there, but the major forces which are, or the dynamic forces which are always there, non-covalent, are the hydrogen bonding, different types of intermolecular forces. I mentioned the van der Waals here, the Casimir forces, other short-range forces, hydrophobic interactions. These are huge proteins containing huge patches of these alpha helical regions containing hydrophobic patches. So interactions between these patches, they make and break due to their changing conformation. And as a result, these forces are handed down from one protein to the other protein till it is a sink where the effector action or the action of the force is elicited in the form of the gene expression, in the form of the differential gene expression. Okay, so I stop here today. The first lecture is over. I thought I would give some time to you at the end for taking some questions. If you have any questions, you can ask me in the chat. And I thank you for your attention. Am I audible? Are there any? Anurag?